And our next speaker was, we were very excited to have him in person, but unfortunately over the last couple of days, this was rendered impossible. So he's Zoom calling from Murcia in Spain. He is Professor Paco Calvo, Professor of Philosophy of Science and Group Leader of the Minimal Intelligence Lab there. He is working at the intersection of plant neurobiology and ecological psychology, and with the support of the US Office of Naval Research, he is trying to study plant adaptive behavior, their intelligence, and how this can inform or inspire biomimetic robots. His recent book, the impressive Planta Sapiens, is part of today's title, Planta Sapiens, Homo Stupidus. And without any further remarks from me, Paco, please take it away.
Hi, let me share my screen. Okay, so can you hear me?
Yes, crystal clear.
Okay, great. Okay, so thanks a lot. Well, first, let me say something, which is like I really envy you. It's a shame not to be able to be there in person. I mean, with you all guys hanging out and in such a, well, let me say, in such a transdisciplinary spirit, because, you know, we are far too used to this talk of multidisciplinarity, interdisciplinarity. But I think what we are having here is something that goes well beyond the capacity of putting a set of tools together, right? So we are kind of trying to raise and answer questions in a whole different way, which has to do with the combination of all these methodologies and toolkits. So in that sense, I really think we are pretty much on the same page. I went through, you know, like looking at the contributors and everything, like, you know, moving from the arts and media, film, and of course, computer science and robotics. And, you know, when I go through the whole set of speakers and range of interest, I really think it's really a shame not to be able to hang out together and spend more time together, right? But anyway, just to give you a brief overview of what is it that we are up to here at the Minimal Intelligence Lab. Certainly one of the things we try to do is, let me just, okay. Yeah, now you can see the full screen, right? Well, first, one thing is that it's a good sign that we consider that plants are as the same as with the rest of the species in the animal kingdom. I mean, so from other kingdoms in the tree of life, that we can truly speak of perception. So these perceptions across the spectrum of life, in the title of your meeting, it really something I really resonate to, right? So that's really good news. Well, I guess from the title, we could remove the question mark and simply say Homo Stupidus, right? So there is something we are truly missing here. Something we are truly missing when we speak of Planta Sapiens, and I think I'll try to convey the general idea of what I mean by we not getting what's at stake here, right? This is what we do basically at the Minimal Intelligence Lab. We combine, if you see the caving here on the left, we combine observations through time-lapse photography. So we do time-lapse photography. We take a picture every minute or so. We assemble the footage, and we are able to observe the overt behavior of plants, right? So that's sort of the idea. Like, let's see how sophisticated plant behavior is by observing the time-lapse footage, and then let's try to make ourselves an idea of whether that type of behavior is sophisticated enough, is adaptive, is flexible, is anticipatory, is goal-directed, for example. So what sort of behavior it is that allows us to speculate that, hey, this is cognition after all. We are truly speaking of plant intelligence or plant cognition, or in this case, plant perception, right? On the other hand, on top of doing the observations through time-lapse photography, we are also doing plant electrophysiology. So if you see on the right, we are inserting electrodes, pairs of electrodes, so that we can monitor the internal activity of the plant in the form of electrophysiological activity, right? So the firing of action potential within the plant body, right? So by combining these two things, observations through time-lapse photography and electrophysiological recordings, we hope to be able to see something that we might be missing otherwise, right? So this would be like a general idea of the setting, like assembling the booths where we do the time-lapse observations. In this case, this is a climbing being approaching a pole, like a climbing to a host pole in this case, and the amplifier to acquire the electrophysiological data that we then analyze. Just for example, just to give you an idea of how different fields in which our research bears upon. Here at the top, you can see the root, the main root of wild rice, and the bottom one is a mutant of this variety of rice. So the top one, you can see that it's circumdotating, is actually revolving as it's moving through differential elongation, and if you see the bottom picture is simply growing in circles. So it looks pretty stupid, right? And the top one is kind of exploring somehow the landscape. So if you think about it, when one type of behavior looks like really stupid compared to the other behavior, which is like more exploratory, bear in mind that all that has changed in between the top and the bottom picture is the fact that the top root can circumdotate, whereas the bottom one cannot. So in this case, we are talking of a system, a plant body, which is fully decentralized, which is non-neuronal. Of course, there are no neurons, no central nervous system. Everything is kind of comes hand in glove with a general that you will be familiar with, embodied, embedded, situated, and active ecological approach. So all this combination, these toolkits of the embodied cognitive sciences, if you see what I mean, they render themselves really useful to try to apply those methodologies to the observation and the explanation of plant behavior, insofar as plants are by necessity fully distributed. They are fully decentralized. So these methodologies that we are used to using in the context of the explanation of animal behavior, they are really handful, or they come really handy when it comes to explaining plant behavior, right? But we can do something else. We can think of how to phyto-inspire our robots. So if you think of this root that could circumdotate and explore its surroundings, and the root that was a mutant that couldn't circumdotate, just look what happens when you try to build a robot out of this inspired in a plant system, right? So this robot is think of this as an inflatable balloon, right? So the actuation is pneumatic, so you are putting pneumatic pressure, so it's kind of like a balloon that gets extended. This can circumdotate, and in that sense, the system can grow and explore the surroundings, right? Like avoid the obstacles. Whereas if you see the other one on the right, it's a system that is not able to circumdotate, and thereby it gets stuck as soon as it finds something. You see, this is the type of decentralized understanding of what I was telling you about. So we can combine the type of embodied cognitive science that we are familiar with the type of phyto-inspiration that has to do, for example, in this case, with the construction of a robot based not on locomotion, like an animal robot would be based on the inspiration of locomotion, like an animal, like locomoting from point A to point B, but in this case, of a robot that grows. We call it a growbot, because it's a robot that grows from point A to point B. So in this sense, we can see that simply by allowing a system that grows to circumdotate, it can avoid obstacles in a way that it couldn't be able to do otherwise, right? So this is one of the things you can do with phyto inspiration for robotics and AI. Now, this is the type of biosensors that we use to measure the electrophysiological activity. And think about it, this is one of the reasons why this is important, is that sometimes through behavioral observations, through time-lapse observation of the overt behavior, we might be missing something that is not really conspicuous to the open eye. So if you get to see through the time-lapse footage what the plant is doing, it might not be that revealing. And in fact, a lot of what's going on, it might be happening underneath. So at the level of electrophysiological activity. So in that sense, if we are able to track, to monitor the electrical activity running throughout the plant body, what we are able to see is that something lights up, something flags at a different time scale. So that's very important, for example, when it comes to agriculture. When you are, if imagine a plant is submitted to some form of stress, could be hydric stress or heat stress, temperature, whatever, any form of stress whatsoever. A form of stress that might not be conspicuous to the naked eye or to time-lapse footage, so you don't get to see it. It's not yet there, the stress manifest, but it is at the level of the electrophysiology. So you might be able to train or to develop an algorithm to interpret the electrophysiological data and see how the plant is anticipatorily taking measures ahead of time to deal with those contingencies before it's too late, right? So that's something that's why we want to combine the behavioral with electrophysiological measurements to see how them both together can tell you something more than by looking at them in isolation, right? And so this is all, you mentioned in the introduction, the book Planta Sapiens. This is something we have discussed at length, but there is something to me that really bothers me. And it's what I call in the book something like, I think we are in a sense treating plants as locked-in syndrome patients, in a sense. Like think of this behavioral level of analysis in which by looking at the behavior, we would see plants simply sitting there, like being sessile, being rooted, doing nothing. And in a sense, we kind of need to open the communication channel. We need to develop some skill to interpret the internal activity of the plant, to realize that the plant is truly alive and there is something going on in there, despite our incapacity to see it through the time-lapse footage, right? So in that sense, that's the way we need to train our eye, or to rely on other techniques of observation to truly understand what's happening within the plant. And the plant may be sentient, may be fully alive in a sense that we are missing simply by watching the time-lapse footage, right? If you see what I mean. So in fact, this is a legume. So legumes fold their leaves at night, so they go to bed. So this is funny because when I speak about this to people, they say, look, if I say that the plant goes to sleep, people say, hey, now we are talking metaphors. You don't really mean to say that. But if you think about it, this is funny because plants, or in this case legumes, they biosynthesize melatonin. So melatonin, the very same molecule that we use and we know puts us back with our day-night cycle. So when people take a melatonin tablet, synthetic melatonin to ease sleep at night and things like this, or when we speak of sleep as DNA repair, right? So any form of life whatsoever, of course, we are all enslaved, and in a sense, we exploit the information that comes in the form of those day-night cycles, under humidity, temperature, light, all those informations, all those regular patterns around the clock is information that, of course, plants have got to exploit somehow as well, right? So circadian clocks, of course, are ubiquitous, and we all rely on the same type of molecular clocks to make sense out of those regularities. So in that sense, we do mean to say that the plant is going to bed. And do plants, when we see that plants are biosynthesizing their own phytomelatonin, it's funny that when we look through the microscope, we don't identify that molecule and think it's the very same molecule that takes place in the animals. So it's the very same molecule, we just fail to miss it, we just fail to see it, and that's why we even have to have the feeling that we need to add the prefix, and we say phytomelatonin. But this melatonin, the very same melatonin, we also employ, right? So in fact, this is even more dramatic. Just think of visuferane. So any anesthetic, so the type of anesthetic that allows you to put an animal to sleep, right? So you can take a plant, literally, you can take this plant, a Venus flytrap, to the vet, and you can just use the same anesthetic that they use in the vet, right? So we've done this. We've taken the Venus flytrap to the vet, and we've put under the glass bell and use the same type of anesthetic, and you can actually see how the plant is put to sleep. So if you think of this, if you see, this is a mimosa, by the way. The mimosa is the one that you don't even need to do time lapse because it falls in our time scale. So you just touch it as here, and you can see how the plant is folding, right? So the plant folds its leaves in real time. You can just record that, right? And what happens when you anesthetize the plant? So when you anesthetize it, this is, hold on a sec. This is, yeah. So this is what happens to the plant when it's fully anesthetized. You see, now you touch it, and it won't fold its leaves. It simply doesn't fold them, right? Now, Just to remind you, we are using the very same anesthetic that you used in the case of a pet when you take it to the vet, right? Now, what happens when you take it, when you allow some time to pass and the effect of anesthesia wears off? Now is after the effect of anesthesia wears off, you can see that the leaves are starting to fold again, if only kind of more slowly. So it's kind of taking longer. It's like, what happened here? I am coming out of anesthesia at a pace, right? So if you see, when you think of plant sentience, it's not so dramatic if you think of it in the very same terms as you think with animals. So if under anesthesia you have lost something temporarily that you recover somehow when you come out of anesthesia, something similar is happening in the case of plants. We just need to realize that it's the very same thing, right? The very same thing, precisely because we are actually the molecular mechanism, the role anesthesia is playing is doing the very same thing in plant and animal cells. Basically, is messing up the trafficking at the membrane. So it's effectively preventing the action potentials from taking place for firing. So the plant is not firing action potentials anymore. So if you see here the recording, here we have the Venus flytrap under the glass bell with the cotton pads with anesthetics. You can see prior to anesthetizing the plant, this is the action potential. So you can see the spike, the very same spike you would see in an animal subject. Then after the anesthesia has taken place, you see the flat line, right? So it's fully anesthetized, right? Then of course you will again see a spike when the plant goes back to normal, right? So well, by the way, this is Natalie Lawrence. This is my co-author, and I owe her so much. So we were very happy here. She put the prose to the book, and we were so happy here to see the book in the windows here. It was in London, Piccadilly. But more importantly, this is something that I'm having such a big headache to trying to convey this message. When it's like when you talk to people, and in this case to the guy at the bookshop, and tell him, look, this is the very same sort of sapiens that we mean when we speak of Homo sapiens. So if you have, thematically speaking, a shelf where you put books on sapient, don't think that Planta Sapiens doesn't belong there. So we mean it literally, right? It's no metaphor. We mean it literally. Now, when you think you don't mean it literally, it's because if you say, look, if we spoke of plant vision, you might be saying, well, if you are literal about plant vision, what do you want me to do? Shall we picture that you are sticking like two eyeballs to the plant, and that's what you mean by a plant having eyes or having vision? And of course, if that's what we thought we would be missing, what's at stake? So being literal and not being metaphorical doesn't mean that we are gonna exploit those broad analogies. We mean it in a different sense, of course. And here is just an example of the way in which, functionally speaking, you might think of the type of structures that could play the same optical role that is being played when it comes to the exploitation of information in the form of images in an animal retina. So if you have in a leaf structure the type of structure that allows in the cuticle, for example, the external part of the leaf, you have the structure that allows you to play the role of a cornea, and in the epidermis, the same role that plays the role of the lens, and in the mesophyll down there, where you have the chloroplasts for the purpose of doing photosynthesis, you have this type of structures that allow you to play the same functional role being played by a retina in an animal model. Then things look rather different, like with the action potential. Oh, we are not being metaphorical. We mean to say literally that plants are being prevented from firing action potentials. The same could be happening here, right? So when we are speaking, for example, of the optical capacities of plants, we mean it in a whole different way. But it goes way more deeper, this idea. In this paper, we did this observational study, this behavioral study on the climb beam being you saw, right? The climb beam being going up a pole. So if you see, this is the video. So you could see, just pay attention to the top view on the left. So you could see the climb beam being circumnutating from the top, you can see. Then we could try to study the pattern of movement is doing as it's trying to approach the pole. So remember, this is a vine and it requires to reach a pole. So this is flexible, goal-directed behavior. You see, when it's approaching the pole, it's like trying to reach it, right? There it goes, almost there. So actually, I'm going to show you one that is even more dramatic. Watch this one. If you see to the naked eye, I can see we are running out of time, so let me speed up. So you see this pattern of motion is going circular, right? So it's circumnating in circles. Look at the picture on the right, on the left, the top picture, right? So here is circumnating in circles. Now you will see that after a while it skips. Now it will skip the second half of the circle and just fish for the pole. Look, now it's going all the way for the pole. You see that? Now it's going backwards, taking impulse and shooting, fishing for the pole, right? Now, that's pretty impressive. When we look at these types of behaviors, we can't help but think that we are missing something, or that we might be missing something, unless we truly think of this in the very same way we speak of the way animals exploit information through optic information, I mean. So think of the type of optic flow that the system generates when it's moving with respect to the surroundings. So this is what you are doing, right? As you are moving across space, you are generating information as you are coupling in particular ways with respect to the environment. So if this is you sitting here, this is the type of information, optic flow you will be generating as objects expand or contract in your retina as you approach those objects, or you move away from those objects, or steer to one side or to the other, right? So we just need to realize that we are treating here the tree as an object and the human subject as the agent performing the activity and generating the optic flow based information, right? So we just need to think in the very same terms, like the tree could play the role of the agent. So we could be thinking of the tree as the agent, right? So in that sense, if you think of the way we are able to generate optic flow information through movement, through locomotion, remember the phyto-inspired robots at the beginning, where you have these rovers or these robots that are able to grow rather than locomote. We are able to create or to generate information based on optic flow through movement. Now, if you are able to create that type of information through locomoting throughout space, through your surroundings, we are simply saying that the very same thing applies here when you are thinking of an object or of a plant moving in space. So when you think of this plant, of the climbing bean, when you think of the climbing bean circumnating around in this way, right? In this way, it's happening exactly the same. So just think of the climbing bean, not metaphorically, but literally as generating those optic flows. Now you see what I mean, that we are not talking metaphors, we are not trading in metaphors, we are speaking literally of the plant generating the type of energy flows that then can allow itself to guide itself in a subsequent pattern of growth, thereby generating or closing the loop in an ecological manner, right? So that's basically the idea of this approach. I can see we are running out of time, so I'm skipping the last few slides and just get in there for the sake of having some time for questions.
Thank you very much. Can you hear me? Yeah. Thank you very much, Paco. I don't know if you could hear the audience clap because the audio from the room might not be coming to you. I have a lot of questions of my own, but my role is to convey questions from the audience. So are there any questions? There's one there that I can see.
Thank you very much for that. Oop, that's a bit loud. That was a really, really fascinating talk. And I think it was very convincing for the idea that, of course, plants have behavior. And do correct me if I'm wrong, but it certainly sounds like you're trying to say that plants perhaps also have experience, that there is something that it is like to be a plant, and perhaps plants are consciousness. plants have consciousness. But one thing that struck me about most, if not all, of the evidence that you presented in this presentation is that it would also apply to microorganisms in terms of responding to photographic information. Example, we of course have microorganisms that have phototaxis. In terms of anesthesia, I believe a lot of microorganisms, if not all microorganisms that have been tested, will also stop behaving when they have anesthesia. And in terms of melatonin, I don't know about that chemical specifically in microorganisms, but there's certainly a huge wide array of chemicals that we see across the spectrum of life, like DNA and RNA, for example. So if you are indeed arguing that this evidence shows that plants could be conscious, would you say that your arguments also apply across the kingdoms of life, and that all organisms, including single cellular organisms, could be conscious? And if you're not saying that, then I would wonder what could you say about plants specifically, as opposed to single cellular organisms, could make them conscious? And what evidence do you have for that, or what has kind of convinced you that that could be true?
Paco, could you hear this, or do I need to convey it?
I'm sorry, can you recap? I was
Yes, I will try to recap. So,
Sorry.
What you study and what behaviors we know from microorganisms. What are the common principles? What are the differences? Can we talk about plant consciousness, microorganism consciousness? Do they converge or diverge at certain very key points?
Okay. Yeah, I would say, in a sense, that the very same principles apply across the tree of life. So I don't think there is anything particular about plants that we couldn't speak of in the rest of the tree of life, so to speak. So put differently, if there is sentience and if there is cognition to be spoken of at all in the tree of life, it will be at the very stump of the tree of life. So where there is life, there is sentience, and that applies to unicellulars, of course. So to me, the reason why we could put them all in the same basket is that the mistake we are making, think of bacterial chemotaxis, and the very same thing happens with plants. We are just, because analytically speaking, we are forced in a sense for experimental reasons and control reasons to focus on one channel of communication, one parameter and one variable. So we might think of bacterial chemotaxis and think of this chemical gradient and then how it's swimming up this chemical gradient to catch the sugar or something. Or in plants, you can think of how to study phototropic behavior and just think of light itself, right? But just look here. This is just a list we compiled of different sources of information that plants are sensitive to. And I say plants, but of course we know plants, and throughout the tree of life it applies equally. Of course, tailor-made. But to me, the take-home message is that we can speak of sentience at the very stump of the tree of life, simply because we cannot speak of life responding on a one-to-one basis to a surface stimulation. Even though we are focusing on light or focusing on this chemical, that's going to be done in the context of the gravity pull and in the context of these other guys around secreting chemicals and in the context of all these other things happening. So in a sense, any form of life whatsoever will have to weigh all those incoming sources of information and provide a response that is not just adaptive or flexible, but globally adaptive. So we construct in the lab these toy versions in which we're able to focus on one channel, and we forget that in trying to make sense of the way they make sense of their surroundings, they need to have this higher level understanding of seeing this all and finding a way to trade off in between all these sources of information. And to me, that's the reason why that higher level perspective, which is to me the entry route to sentient, has got to be there in the first place. Because first, it's got to be a response that is, as I said, globally adaptive. And second, it cannot be reactive. It's got to be truly proactive. So it cannot be simply waiting for this information to impinge on the sensory surface and provide a response reactively. They've got to respond anticipatorily, and they can only do that if they have this sense of valence built on top of those individual one-to-one signaling systems, right? So in that sense, I would say that the same predicate, so think of habituation, just to use a predicate from the learning literature. So that mimosa that you saw being anesthetized, mimosa is a plant that learns So habituation is a primitive form of learning, sure, but it's the very same type of learning, habituation, that we can try experimentally on any other form of life whatsoever. And we will know that it's been tried in unicellulars, and we can speak. So we will just have to customize the protocols, because the time scales will be different, because electrophysiological speaking, again, the time scales for the electrophysiological signaling will be different. But despite those idiosyncrasies in terms of the time scales where those behaviors unfold, there is something that unites us all, and it's the sense in which we need to make sense of the surroundings in a proactive, anticipatory, and global adaptive way.
Thank you very much, Paco. It would be great to have you here and continue this lovely conversation over a coffee break. But at this point, let's thank Paco once again.
Thank you.